Right. Hello, everyone. In this session, we'll be looking at what happens when things go wrong in red blood cells. So we'll be looking at the pathologies of erythrocytes. Okay. So in order to do that, what we'll be doing is looking at these learning outcomes. So in order to determine whether something has gone wrong, we, as a clinician, as a person who looks at blood tests, needs to have a reference range. Okay, so we'll be looking at reference ranges, and it's this that's used as a context to determine if something is beyond that reference range or something is below that reference range, and that indicates whether something is wrong. So we'll be looking at anemia mostly in this session. Okay, so we'll be looking at anemia, but at the end we'll also talk about too many red blood cells, or polycythemia. Okay, and also talk about we'll talk about anemia to be in a bit more detail. Okay, and we'll look at the reasons why, or the two main reasons why anemia develops. Okay, there are many reasons, but there are two main reasons. Okay, and that's the role of iron, okay, or iron deficiency anemia, and also pernicious anemia, okay, which is a measure of intrinsic factor, but also it could be a measure of vitamin B12 and folic acid. Okay, and then we'll also talk about sickle cell anemia, aplastic anemia as well. And then we'll also have a little look at the difference between polycythemia and endocytosis. Okay, polycythemia is obviously when there are too many red blood cells, and which could be a normal reaction. Okay, it could be a normal response your body makes, but usually it's something there's something that's gone wrong. Okay, another word that's often confused with polycythemia is endocytosis. Again, this is something that happens normally. Okay. Um, okay. So, okay. Step back and just refresh our memories on the importance of red blood cells, or erythrocytes. Okay. Now, with red blood cells, the main function of them is to bind oxygen. Okay. Bind oxygen to the hemoglobin in red blood cells and take that oxygen to every cell in the body that requires oxygen, which obviously is every cell. Okay. Oxygen is important because it's required for aerobic respiration. Okay. If you remember from the biochemistry lectures that we had, oxygen plays a role as the final electron acceptor in the electron chain pathway in the mitochondria. Okay. And without that, we can't get the electron chain pathway working properly. So we need sufficient amounts of oxygen to ensure that enough ATP is being produced by the cells. Okay. Um, ATP is obviously the energy molecule that other biochemical processes in the cells use, and without ATP, those biochemical processes can't be carried out, and because of that, cells, if ATP deprivation goes long enough, then cells will die. Okay, so it is a matter of life and death. Okay, so in order for every single cell, because if you think about it, there are trillions of cells in your body, okay, and each one of them requires oxygen all the time, okay, and so your body then has to maintain, in order to ensure their survival, your body has to maintain erythrocyte number, or red blood cell number, at the correct levels. Okay, it also has to maintain that the erythrocytes function properly as well. It's not only their number, but also their shape, okay, and how they function. Okay, so we do have this a system in our bodies called homeostasis. Okay, and this ensures, amongst many other things, but it also ensures that the number of red blood cells in our bodies remains correct at all times. Not too few and not too many, just right. Okay. And from the other lectures that we had so far, we know that that the process of making red blood cells is a complex process. Okay, it's a multi-step process which requires a multitude of genes. Okay, but not only that. Okay, it also requires sufficient levels of uh, of nutrients. Okay, iron, vitamin B12, folic acid, all of these things have to be in sufficient quantities inside your body. Okay, so if there's a problem with your genes, okay, or if there's insufficiency in your diet, then that can lead to problems. Okay, red blood cell problems. Okay, the easiest thing to fix is usually the dietary. Not usually, it is the insufficiency of nutrients in your diet. You take those diets, and usually the problem is fixed. Okay, bigger problem is when you have a problem with uh, with your genetic regulation of this process. Okay, have a look at reference ranges. Okay, so when you go to your GP, when you go to your doctor, and they suspect something, they'll extract some blood and send it for a blood test. Okay, if they suspect it's something to do with red blood cell count, or maybe they suspect you have anemia, okay, what they'll do is they'll do a blood test that consists of several um, analyses. Okay, so these are the types of parameters that they measure in a blood test if they suspect anemia. Okay, now they have a reference range by which they go by. Okay, so everything should drop between these ranges. Okay, and if anything's below or anything's beyond, it's an indication that there needs to be a follow up. Doesn't necessarily mean that something is wrong. Okay, but it needs to be followed up. Okay, so the simplest one is hemoglobin. Okay, so how much hemoglobin we have in our cells, and it's usually a measure of total hemoglobin inside our in our blood tests. Okay, it's measured in grams per liter. Okay, so 139 grams per liter of blood should be hemoglobin. Okay, another one is mean cell volume. This is a measure of the actual physical uh, properties of your red blood cells. Okay, mean cell volume. This is what the reference range is. So your volume of every red blood cell should be between 79 to 99. Okay, in this typical result, it's about 87. It falls between that, so everything is normal. Okay, F stands for femto. Okay, so if you remember, so we have milli, micro, nano, pico, femto, and each time it's going down by 1,000. Okay. So red cell count's clear. Okay, it's just basically the number of red blood cells per unit volume. Okay, mean cell hemoglobin. Mean cell hemoglobin. This is the sorry, these are the units by which we measure. So it's picograms per cell. So 30.3 grams in this typical result. Now, it obviously falls between this range. So this whatever this result came from is considered normal. If there is lower, if the mean cell hemoglobin is lower than the reference range, that indicates an anemia. Okay. Now, when they measure or when they make an initial diagnosis, they won't just use one parameter. They'll use the whole range of parameters to give a diagnosis if there is a problem. Okay. So another parameter that they measure is mean cell hemoglobin concentration. Okay, whereas this mean cell hemoglobin was per cell, this is mean cell hemoglobin concentration is per volume. Deciliter is 100 milliliters. Okay, so 347 grams per 100 milliliters of blood. Okay. As far as the mean cell hemoglobin concentration is concerned, okay, the range can be low, normal, or high, with respect to the reference range. Okay, and the red blood cell number could be the same. Okay, it could be normal. So if it's high, if the concentration is high of hemoglobin and the red cell number is normal, okay, it could still be quite normal. Okay, usually if the concentration is low, okay, and the red cell count is normal, it usually means that the actual phenotype of red blood cells, so the physical appearance of red blood cells, are much smaller, so they tend to be microcytic. Okay, so obviously that means there's something wrong. Okay, hematocrit levels, so that's just the blood, that's just a fraction of red blood cells in the whole blood volume. Okay, reticulocytes, so these are the immature red blood cells that get released from the bone marrow, and these give an indication of how well erythropoiesis is happening in the bone marrow. What you normally have is this range here between 25 to 125. The range is quite big, but this is considered normal. Okay, so this falls within the normal range, so everything is normal here. Okay, if there aren't enough, if they're less than 25, okay, it could be that erythropoiesis isn't happening properly. That could be through many reasons. Okay, it could be a genetic problem. It could be dietary insufficiency. Okay, or it could be that they're being mopped up and destroyed once they get into the bloodstream. Okay, so there could be a few reasons. Okay, the next one is red cell distribution width. Okay, this is really a measure of the variation in volume and size of the red blood cells. Okay, normal red blood cells are about 6 to 8 micrometers in diameter. Okay, now if they look at your red blood cells and they see that there's quite a big variation there, so there's lots of cells which are smaller than 6 to 8 micrometers, and there are some cells which are bigger than 6 to 8. That means the distribution width is big. Okay, and that means there's something wrong perhaps, and it needs to be followed up. And the next one is erythrocyte sedimentation rate. This is an indirect measure of inflammation, of systemic inflammation in your, well, basically in your blood. Okay. Now, inflammation is something that's important. It's necessary. When you have a wound, inflammation is necessary to get that all fixed up. But the inflammation is localized to the wound. Okay. But there are some conditions where if your body is stressed, you suffer from systemic inflammation. So it's inflammation throughout the body. And so if there's lots of inflammation, if there's systemic inflammation, it has an impact on how quickly your erythrocytes sediment, okay, or drop to the bottom in a tube. Okay. So and one of the effects, one of the impacts of inflammation is the upregulation of something called fibrinogen in your blood. Okay, fibrinogen is a precursor to fibrin, which is the sort of meshwork that's produced when your blood clots. Okay, so if there's lots of fibrinogen around, it could indicate that there's more inflammation, more systemic inflammation. Okay, now red blood cells, they don't normally stick to each other, and because they don't stick to each other, they fall at a particular rate. Okay, if there's lots of fibrinogen knocking about, the red blood cells will more likely be attached to that fibrinogen. Okay, and then other red blood cells will attach to the fibrinogen as well, and they'll clump together. And if they clump together, they're bigger, they're heavier, so they sediment faster. Okay, so if they sediment faster, that means um, there's probably some kind of inflammation, systemic inflammation happening. Okay, if there's inflammation, okay, the plasma viscosity also increases as well. Okay, so the more fibrinogen you have floating around, the more viscous the plasma is. Okay, so it's to note that we're talking about plasma here, not whole blood, but just plasma. And fibrinogen is dissolved in plasma. So the more fibrinogen you have, the more viscous the plasma becomes. Okay, so these are the kinds of things that they would test in a blood test. There are other things as well, okay, and which we'll talk about later on. Okay, so to the next slide. Okay, so just to summarize, then summarize your understanding of anemia. Anemia is anything that falls consistently below the reference range. Okay, so and we're looking at the hematocrit, so the red blood cell portion of whole blood. Okay, so if it's consistently lower than what is considered normal, and then we know that it's some kind of anemia. Okay. On the other hand, if your hematocrit levels are consistently higher than the normal range, okay, and then it could indicate polycythemia. Okay, I say indicate because you can't say for definite whether it is polycythemia. It could be erythrocytosis. It could be a natural response by the body to increase the number of red blood cells, and so that's why I have to use the word consistently. So, okay, so when you do multiple blood tests, and it's always the same. Okay, and when I say multiple, I'm talking about when you're doing it maybe a week after, maybe a month after. Okay, and they're still quite high, and then it could indicate polycythemia, and that could indicate some kind of disease. Okay, right then. Okay, now how do we develop anemia? Okay, or polycythemia? Okay, so how how something arises as far as disease is concerned, we refer to that as the etiology of the disease, or how it comes about. Okay, so if we're looking at iron or vitamin B12 deficiency, okay, so these are the kinds of the building blocks, if you like, of manufacturing red blood cells. So if they are lacking, okay, as well as folic acid, then what happens as a consequence of manufacturing red blood cells is that the erythrocytes, the red blood cells, look really small. Okay. On the other hand, so if they look really small, usually the number of red blood cells looks normal. If you look at them individually, they look normal in terms of the numbers. But when you look at them under a microscope, you see that they look much smaller. So in terms of the numbers, they look normal, but in terms of the shape, they look really small. On the other hand, you could have few red blood cells, but they could look really big. Now, either if they look really small or they look really big, they are not functioning properly. Okay. Um, they need to be 6 to 8 micrometers in diameter, and they need to have that semi-concave shape. Okay. For them to work properly. Okay. Now, the next one is a problem in tropical countries where malaria is endemic. Okay, as you know, some of you may know that the life cycle of the malaria pathogen parasite. Okay, part of its life cycle happens in the red blood cells, so it sort of gets into the red blood cells and reproduce there, and in the process of doing that, your red blood cells become useless. Okay, and so therefore they rupture. And so the malaria parasite, in order to fulfill its life cycle, it destroys your red blood cells. Okay. Another problem could be your own immune system. Okay, so an autoimmune condition whereby it attacks your red blood cells. Okay, in some form. So it could be a precursor of your red blood cells. It could be the reticulocytes. It could be the red blood cell, the fully functioning red blood cells themselves. Okay, there's a whole process of maturing red blood cells in the spleen, and it could be that at this particular site in the spleen, your red blood cells are destroyed. And these things happen. It's very rare. Okay, and in those people whose spleen whose maturation of red blood cells in the spleen causes them to be destroyed, and therefore the number of red blood cells decreases. In those people, the spleen is usually removed. Okay. But anyway, as a consequence of of malaria or autoimmune conditions, okay, those cells are those red blood cells are destroyed, so the number of red blood cells decreases. Then, so the total red blood cell count is a lot lower. Okay, physical loss of cells due to an external cause could be a hemorrhage. Okay, could be. It doesn't have to be a hemorrhage. It could be a chronic bleed. So where there's a bleed that's small but it's happening constantly. Okay, and this will impact on obviously the number of red blood cells, and also the hemoglobin levels. Okay. Uh, Okay, so there are intrinsic defects or genetic defects that can cause your hemoglobin to not be produced properly or manufactured properly. Okay, the most common type is sickle cell anemia or sickle cell disease. Okay, it could also be that if you remember, the process of manufacturing red blood cells is quite a big multi-step process requiring different enzymes and different processes. If anything goes wrong with those processes or those enzymes or those genes coding for those enzymes or those genes coding for those processes, okay, that can lead to red blood cells that just don't function properly. Okay, so because it's usually a genetic problem, we call that intrinsic. Okay, as opposed to extrinsic. Okay, so in this case, red blood cells are they can be recognized as being abnormal and can be eliminated. Okay, so sickle cell disease, they're not so much eliminated; they have a lot of lifespan, so they are eliminated quicker, but they're not eliminated immediately. Okay, there is a whole process of maturing red blood cells to make sure that they all work properly. And if they don't work properly, it's a target for your immune system to eliminate them from the system. Okay. Okay. So stress. If somebody is stressed or your body is stressed, doesn't have to be emotional stress. It just can be physical stress or attacked by a pathogen. Okay. And that attacks the bone marrow. Okay. And it could be the side effect of a drug. Okay. If it's a side effect of a drug, perhaps somebody has cancer. Okay, and so the chemotherapy drugs usually target those cells which are reproducing really quickly, and your bone marrow cells are one of those cells types that reproduce really quickly. So they're a non-specific target for a cancer that perhaps is happening in the lungs. So because of this non-specific uh, inhibition of the bone marrow cells, and because blood is made in the bone marrow, that can impact on your red blood cells. So chemotherapy could impact on your red blood cell number diminishing. Okay. So problem with the regulation of red blood cell production. So again, this is kind of refers to the intrinsic defect. Okay. Um, uh, it could be an intrinsic defect, not so much to cause a decline, but more so to increase the number. Okay, so if there are many processes which have negative feedback loops. So your body tries to increase something, and then when the number is reaches a level, there's a negative feedback loop to say to the cells to stop doing that now. There's enough, so stop it. Now that's a regulatory process. So if something happens in that regulatory process, there's no negative feedback loops. There's no negative feedback loop rather, and the numbers continue to rise. Okay, so in this case, we could have more red blood cells. Okay, and as a consequence of that, your whole blood becomes thicker. Okay, and it has a sort of a sludgy consistency. Okay, and if that's something that's consistent, okay, after many blood tests, then it could indicate malignancy. Okay, okay, and not enough oxygen in the tissues, hypoxia. Okay, this is completely normal. People suffer from hypoxia all the time. If you're exercising, there are certain your muscles will those muscles which are being actively used, they're going to suffer from hypoxia. Okay, and so therefore your red blood cell number will increase in response to that. Okay, again, blood will be too thick. Okay, and it could indicate polycythemia. Okay, but it's more likely erythrocytosis. So it's a natural response to hypoxia. Okay. Okay, so as far as anemia is defined, there's two ways you can define it. You can have a functional definition or you can have an operational definition. Operational is more about uh, clinicians who work in the lab, and functional definition is something more. Um, it's more the effect on the body. Okay, so anemia, the functional definition is that there's a decrease in oxygen carrying capacity of blood, and this could be because there's insufficient hemoglobin, or the hemoglobin, there's something wrong with the hemoglobin, so it's impaired. Okay, usually what's more common, if you look at the population as a whole, is that usually it's the hemoglobin which is insufficient. Okay, rather than impaired. So impaired hemoglobin would be sickle cell, for example. Now, one operational definition would be a reduction in the hemoglobin content due to decreases in erythrocytes. Okay, so that's after a blood test. So a clinician would look at the blood test and say, "Oh, there's a reduction in the hemoglobin content." Okay, due to a decrease in the erythrocyte number. Okay, as well as the hematocrit fraction being smaller. Okay. So that's those are the two types of definitions that you might come across. Okay. Now, people who have anemia won't necessarily know they have anemia, not for a long time. It's because the anemia, the reduction could be quite small to begin with, and it just builds up as you age. But because it's so small in the beginning, you don't really notice it, and you don't notice it because your body has this ability to adapt to this reduction. Okay. Now, if the blood loss is severe and it's sudden, which could be the case if somebody suffers from a hemorrhage. Okay. If it's severe like this, that can lead to drastic effects. Okay. It can lead to your blood pressure dropping drastically low. Okay. Where you get severe hypotension, and if it's really severe, your body can go into shock. Okay, that's because just there's simply not enough blood. There's simply not enough red blood cells to carry enough oxygen to perfuse all your cells of your tissues and organs. Okay, so if that's the case, then your brain won't receive enough oxygen, your heart won't receive enough oxygen, your liver won't receive enough oxygen, your lungs, etc. Okay, and these are vital organs. Okay, so in the immediate aftermath of a hemorrhage, okay, your body will try to adapt by sort of redistributing blood flow to the vital organs, such as the brain, the liver, the heart, and the kidneys. Okay, these are considered the vital organs. This is where any drop in oxygen delivery can have drastic um, effects. Okay, so that's the importance of blood, and anemia um, can lead to hypertension. Okay, and if you have hypertension and you're exercising, you might feel that the your hands become numb, for example. It's because your body is shunting blood to more important parts of the body, such as the brain, and the heart, and the liver, etc. Okay, so if your hands are becoming numb when you exercise, it might be something to consider to go to the GP and get yourself checked. Okay. Right then. Okay. One of the physiological adaptations that your body utilizes or takes advantage of, okay, is using this molecule here. Okay, 2,3 biphosphogylycerine. Now it's a molecule that you might remember from biochemistry. Okay. This molecule is actually a modified form of this molecule here, 1,3 biphosphogylycerine. Okay, this is the molecule that's often found in glycolysis. Okay, glycolysis happens in red blood cells. The red blood cells have to manufacture some kind of or some level of energy, and that happens during glycolysis in the red blood cells. They don't have mitochondria, so we don't have the electron chain pathway, but the net two ATP molecules that are being produced by your red blood cells is sufficient. Okay, but getting back to the point, one of the molecules in the pathway of glycolysis is this biphosphoglycerine. Okay, so the breakdown of glucose eventually leads to the production of 1,3 biphosphogylycerine. Okay, and this is a slide that I've taken from one of the biochemistry lectures. Okay, this is the process of glycolysis. You can see it being broken down, linearized, and sort of broken down and modified. And so this is 1,3 biphosphogylycerine. Okay, and so this 1,3 biphosphogylycerate is converted to 2,3 biphosphogylycerate. And the thing with this molecule here is that it actually binds to hemoglobin. Okay, and it binds to hemoglobin. It actually binds to deoxygenated hemoglobin with greater affinity than oxygenated hemoglobin. Okay, now that's important to remember or to know, because when BPG or 2,3 biphosphogylycerate binds to O2 or binds to hemoglobin rather, O2 simply doesn't bind as tightly to the hemoglobin. Okay, so if you consider your red blood cells with this oxygen bound and they're being delivered to cells, okay, until there's a certain level of acidity in the red blood cells, the oxygen doesn't come off. Okay, so to adapt to that, okay, BPG will bind to hemoglobin and it'll make the oxygen come off a bit easier. Okay, and because it comes off a bit easier, the person who has anemia won't feel the symptoms of anemia, so they'll be asymptomatic. Okay, because they don't feel the symptoms, there's never a need to visit the doctor, and so it never gets discovered. Okay, so this is one of the adaptations or physiological adaptations that your body uses or takes advantage of to mitigate against anemia. Okay. Okay. So in response to anemia, okay. So anemia might cause hypoxia every now and then, and in response to hypoxia, your kidneys will then produce erythropoietin because that blood which has insufficient levels of oxygenated hemoglobin will eventually reach the kidneys. Okay, the kidneys will detect that and produce and release erythropoietin. Okay, into the bloodstream. Okay, then that then gets to the bone marrow, and that stimulates the bone marrow to produce more red blood cells. Okay, now once those red blood cells are produced, okay, we know that red blood cells usually live for about 4 months, about 120 days. Okay, and usually on a day-to-day basis, we're losing around 1% of our blood every day. Okay, that's being so 1% of blood is being lost every day. Okay, so that 1% that's being lost has to be replenished in order for your body to meet the demands of oxygen delivery. That 1% that's lost has to be replenished. Okay, by erythropoiesis. So we need the kidneys to be constantly working to release the right levels of erythropoietin to get the right levels of erythropoiesis. And usually, if given everything is normal, and then everything works fine. Okay, but effective erythropoiesis requires, as I've alluded to before, sufficient nutrients such as iron, vitamin B12, and folic acid. Sometimes it says folate, or sometimes it says folic acid. It's the same thing. Okay, so folic acid and folate, the same thing. Okay, so it requires uh, sufficient amounts of these nutrients, but also it requires that your genetic regulation for ensuring the processes that produce red blood cells work properly as well. Okay, so it requires both of these to be working properly. Okay, so anemia, okay, can be due to ineffective erythropoiesis, or it can be insufficient erythropoiesis, and this is kind of a reflection of whether there's an insufficient amount of nutrients, or whether there's something wrong, or whether there's a genetic problem with the processes that produce red blood cells. Okay, so ineffective erythropoiesis. So if you're lacking vitamin B12, lacking folic acid, okay, both of these are essential for DNA synthesis. Okay, we'll go and talk about DNA synthesis and the relationship to vitamin B12 in a few minutes. Okay, but that leads to megaloblastic anemia, where the red blood cells look really big, but there's not enough red blood cells, so the red blood cell count has dropped. Okay, so um, another feature of ineffective erythropoiesis is globin synthesis. Okay, so remember, hemoglobin, heme is one part and globin is the other part. Okay, and so if there's a problem with globin synthesis, that can lead to another condition called thalassemia, which is a form of anemia. So because you don't produce globin, or you're not producing enough globin and enough heme, okay, you're not producing enough hemoglobin. Red blood cells require hemoglobin, so if you're not getting sufficient amounts of hemoglobin in red blood cells, those red blood cells are ineffective. Okay. And another feature could be the heme portion of hemoglobin. Remember, the precursor to the heme of hemoglobin is protoporphyrin. Okay, so if your body has defective genes that doesn't produce enough protoporphyrin, okay, that's protoporphyrin anemia, and that's its technical name is sideroblastic anemia. Okay, so whatever the reason is, okay, the the number of erythroid precursors, so the cells before reticulocytes, those are all erythroid precursors. There's simply not enough of them being produced. Okay, and not only are there not enough being produced, but whatever is being produced are defective. And if they're defective, your body, your immune system will recognize that and trigger their death. Okay, so apoptosis of those erythroid precursors, because they're not going to work. So there's no point them consuming energy. So best to destroy them and wait for some good fresh ones which do work. Okay. So apoptosis, so cell death of those erythroid precursors, that will obviously mean that there's a drop in reticulocytes in the circulation. Those reticulocytes develop and mature into red blood cells. So this, because you don't get enough of the reticulocytes, you get a drop in red blood cell number and therefore anemia. Okay, insufficient erythropoiesis, again, it could be to do with the amount of iron intake, okay, or your body's inability to absorb iron properly. Okay, it could be that there's lots of that there's not so much lots of bleeding. There could be chronic bleeding, which means that there's a bleed somewhere in your body that's bleeding all the time. It's not being stopped, so it's chronic. Okay, kidney disease. Okay, because kidneys are responsible for producing erythropoietin. Okay, so all of these, so a loss of not enough iron, iron not being absorbed, not being processed in the body properly, chronic bleeding, kidney disease, all of these can lead to insufficient erythropoiesis. Okay, so it's just simply the quantity of erythropoiesis isn't happening what it needs to be. Okay, so that obviously leads to a reduction in erythroid precursors. Okay, because it's insufficient, that drops the number of red blood cells in your blood, and that can lead to full-blown anemia. Okay. Okay, so just to summarize there then. Okay, ineffective erythropoiesis, driven mainly by erythropoietin, but other growth factors play crucial roles. Now, I've mentioned that because I've talked about erythropoietin, but you must remember there are other chemicals, other molecules that interact with erythropoietin. Okay, um, that also ensures that we're getting the right amount of erythropoiesis happening. Erythropoietin is by far the most important molecule. Okay, but other molecules do also play roles, and so just because you have enough erythropoietin being produced, if the other substances, other molecules aren't being produced at the right levels or at the right times, and that can also not inhibit, but it could drop the process of erythropoiesis from happening at the rate it should be. Okay, and that can lead to the erythroid precursors being defective if there's a genetic problem. Okay, and that can lead to their death then. Okay, so the types of anemia that we've talked about so far, megaloblastic. This is usually because we don't really have enough vitamin B12 or folic acid in our diet, and that has an impact on DNA synthesis, and that can lead to megaloblastic anemia. Thalassemia is when your body doesn't produce enough globin molecules, so hemoglobin. So the globin element of hemoglobin isn't being synthesized properly. Okay, that is a thalassemia. Sideroblastic. This is where the precursor to the heme element of hemoglobin, the protoporphyrin, there's deficiency in its synthesis. Remember, in the one before that, actually, the lecture, when I was talking about red blood cells, we talked about the process of protoporphyrin production, where it goes into the mitochondria, then it goes out the mitochondria, then it goes back into the mitochondria, and it requires several enzymes, okay, to produce functioning protoporphyrin. So the production of protoporphyrin, again, just in itself is quite complicated. Okay, so the idea really is to sort of make you appreciate or help you appreciate that anemia, okay, a simple drop in red blood cell number, there could be several reasons behind that. It's not a simple case of just looking at one possible reason. There could be several reasons. It could be genetic, and if it's genetic, it could be any gene that's responsible, that plays a role in the process of erythropoiesis. Okay, or it could be something simple such as insufficient vitamin B12, iron, or folic acid. Okay. Okay, insufficient erythropoiesis. So simply, there's just simply not enough happening. Okay. So the one before, when we talked about ineffective erythropoiesis, that's usually a problem of genes. Okay. Whereas this is more a problem of the environment. So not enough iron. Okay. Not enough iron. Not enough vitamin B12 or folic acid. Okay. So it could be that the intake is low. So if you're, if somebody is vegetarian, doesn't have to be vegetarian. Okay, it could, but vegetarians suffer from iron deficiency much more than people who eat meat. Okay, that's not to say that there aren't vegetarians who don't suffer from iron deficiencies because they supplement their diet with iron-rich foods such as spinach and green leafy vegetables. Okay. Okay. There could be a problem with the absorption of iron. Okay, we'll talk about the absorption of iron in a few minutes, but that in itself, actually we talked about that before. We talked about iron absorption in a previous lecture. Okay, um, and excessive blood loss. So um, there could be a polyp or some kind of outgrowth in your colon, in your small intestine. That could be bleeding ever so slightly, but it's nevertheless bleeding. And that blood then is lost in the stools, and it can be detected in the stools. Okay, somebody has renal disease. Okay, that can lead to a deficiency of erythropoietin production. Okay, so remember this is the stimulant for erythropoiesis. So if you don't get enough of this erythropoietin, there's just not enough erythropoiesis being stimulated. So and that can lead to a drop in red blood cell number. Okay, aplastic anemia, a destruction of the erythroid precursors, and this could be due to an autoimmune condition. Okay, so it's your own immune system producing antibodies towards the precursors or even the stem cells that make up red blood cells. Okay, and another reason could be cancer. Okay, not necessarily cancer of your bone marrow. Okay, but it could be a cancer somewhere else. It could be a lung cancer. Okay, so those lung cancer cells have come off from the lungs, entered the blood circulation, then transported and delivered to the bone marrow, and then they basically outgrow the bone marrow cells. Okay, so they take over, and those malignant cells that originated from the lungs have no property to produce you know red blood cell precursors. Okay, so there's lots of reasons for insufficient erythropoiesis. So here's a little diagram, a picture just to show you the inside of a large intestine where somebody is suffering from polyps. Okay, so it's an outgrowth from your large intestine, and you can see that it's constantly bleeding. Okay, so we're losing blood, and this can be detected. Sometimes the stools look red, or there's a smear of red. Okay, or it could be that the blood has been broken down in the process of the stools being produced, but that can be detected in the stool sample. Reason for anemia could be the breakdown of red blood cells. Okay, so the rupture of red blood cells. Okay, and that's hemolysis or hemolysis. Okay, and you can see from these test tubes here, this is a normal blood sample. This is the hematocrit at the bottom, plasma here, and this is what it should look like. It should look yellowish. Okay, but if the red blood cells rupture, okay, it can release the hemoglobin from the red blood cells into the plasma, making the plasma look reddish. Okay, and so there's different degrees of rupture of hemolysis. Okay, they can be caused by different things. there could be intrinsic defects. There could be problems with your own genes. It usually is a problem of the erythrocyte membrane. So the erythrocyte membrane is a specialized membrane. But if the membrane is defective, okay, or the enzyme systems that help produce the erythrocyte membrane are defective, okay, or the hemoglobin itself is defective, okay, as in the case of sickle cell anemia, okay, and even thalassemia as well. Okay, that can cause your membrane to be less stable. Membranes of your red blood cells to be less stable, and cause their lysis. Okay, so as opposed to intrinsic defects, there's extrinsic causes as well. So autoimmune conditions where your own antibodies target red blood cells. Okay, your red blood cells are targeted naturally by your immune system, because your immune system is constantly monitoring red blood cells, to see how effective they are. If they see that they're not really effective, they're removed from circulation, and there's a process by which that happens. Okay, so and that process also involves hemolysis. Okay, so it could be your own antibodies, your own immune system causing hemolysis. Okay, there's another component in your blood called complement. Okay, that can also bind and target the membranes of red blood cells. Okay, bacteria have toxins which bind to red blood cells and can cause their rupture. Okay, um, okay, so there's intrinsic and extrinsic causes of hemolysis. Look at iron deficiency anemia. Okay, so basically anemia, a drop in red blood cells that's caused because there simply isn't enough iron in your system, in your bodies. Okay, so when you consider how much iron is lost on a daily basis, okay, we need to consume about a milligram of iron every day, okay, to to counteract the effect of the 1 milligram that's lost. Okay, where if you think about your skin being your skin is being shed every day, your skin cells will have iron in them, so that's lost in the environment. There's cells in your mouth which are being shed. There are cells in your all along your digestive tract which are being shed, and every cell will contain a small amount of iron. So when you take all the cells which are lost, okay, and if you measure the amount of iron that's lost as a result of that loss, it amounts to about 1 milligram. So if your body is working properly and your genes are working properly, the genes responsible for iron metabolism and iron absorption and red blood cell production and erythropoiesis. Okay, if everything's working properly, we need to replenish that 1 milligram from our diet. Okay, sometimes we need to take more than the 1 milligram. Okay, because if children are growing rapidly during their infancy stages. Okay, so we need more iron in their diet. Okay, so during infancy, childhood, and adolescence, the child is still growing, and they're growing quite quickly. Okay, so we need a bit more iron. That means, okay, if a woman is pregnant and she's producing milk, that milk is supplemented with iron. So your body shunts some of the iron stored in your bodies into the milk. So the milk is enriched with iron to feed the baby. Okay. Impaired absorption. So people who have gastrointestinal problems, okay, such as celiac disease, okay, that can cause iron to be absorbed to not be absorbed properly, okay, or it can cause inadequacy in absorption. Okay, another molecule which is produced by the liver called hepcidin. Okay, it's an antibacterial property, but it has other properties as well. It stops iron from entering the circulation. So if your liver is producing lots of hepcidin, okay, it prevents iron that's stored inside cells from being released into the blood circulation. Okay. Reduction in acidity of the stomach. Okay, this is important. Your stomach needs to release a certain quantity of acid, and normally everyone's stomach releases the right amounts of acid, but sometimes they don't. Okay, in some people that doesn't happen adequately. It could be because of medicines. Uh, Medicines that they take, so some of the side effects of medicines affect the stomach, okay, and the stomach's capacity to release the correct amounts of of acid. Okay, the acid release in the stomach is important because it helps to convert the non-absorbable form of iron into the absorbable form of iron. Okay, so ferric is non-absorbable, whereas ferrous is absorbable. Okay. Okay, and then the last one, chronic blood loss. So, like I was showing before, the polyp that was constantly bleeding. Okay, it doesn't have to be a polyp. It could be ulcers in the intestine, in the stomach. Okay, and they can be caused by people who mismanage their diet in terms of their medicines they take. If they maybe take too much aspirin, okay, or they consume too much alcohol. All of these things can potentially cause gastritis, inflammation of the stomach, and that will prevent your stomach responding properly. So it won't release as much acid, perhaps. Okay. Okay. So yep, tumors, hemorrhoids. Okay. So blood vessels in the large intestine, in the rectum. Okay. Okay, and heavy menstrual bleeding, fibroid tumors, all these things can be causes of chronic blood loss. Okay, and because iron, because your blood is recycled, um, and the iron is recycled as well. Um, if your iron is being lost because you're losing blood, that iron is then not is can't be recycled, so it's lost. Okay, so it's something else to consider. So chronic blood loss prevents that iron from being recycled, so you're having to consume more iron to make up for that loss. Okay, so now in terms of looking at iron deficiency anemia. Okay, your body, because I did mention before in a previous lecture that iron is a dangerous substance. It's very important for our good health, but it's very reactive. Okay, it participates in lots of reactions. It can accept electrons quite readily, and it can give away electrons quite readily. Okay, and because of that, there are other chemical process, other reactions happening that can take advantage of that. Okay, and if there's a reaction that freely donates and accepts electrons, that can lead to the production of free radicals and reactive oxygen species, which are quite toxic molecules. Okay, so to prevent that from happening, iron is usually complexed with other molecules for safe storage. So inside cells, iron is stored in complex with a protein called ferritin. Okay, inside cells, it doesn't have to be. It can be inside bone marrow cells, the liver cells, macrophages. Okay, but any cell that has iron stored is usually stored as ferritin. Okay, there's transport iron. This is the protein that's complex with iron once it enters the bloodstream. Okay, so transferrin is something that's also measured in your blood test. if somebody suspects anemia. Okay, ferritin again is it's a molecule that's measured in your blood test as well as transferrin. Okay, so so our storage iron and transport iron. Functional iron is classified as that iron which is performing the function. Okay, so the function of iron in complex with hemoglobin is to carry oxygen. Okay, so in this case, it's basically the iron that's inside red blood cells. Okay, inside red blood cells complex as hemoglobin. Okay, inside other cells, myoglobin and other important molecules, cytochromes. Okay, so iron is complex with any of these. Okay, and all of these perform vital functions, so we refer to that as functional iron. Okay, now in terms of storage, where would you find most of the iron? Okay, now most of the iron that's stored actually is in the functional compartment. Okay, so the functional iron, that's where most of the iron is. Okay, so hemoglobin in the iron, myoglobin in the muscles, okay, and then other important enzymes in many different types of cells which are also complex with iron. So peroxidase, catalase, these are the enzymes which need iron for them to work. Okay. The second biggest component is storage in the cells. Okay, storage in other cells, not so much red blood cells, but in other cells. So okay, so iron stored in complex with ferritin inside cells, and then the least populous of iron storage is the transport iron. So that iron that's complex with transferrin. Okay, so these are the three main compartments where you would find most of the iron. Okay, now when we look at anemia, okay, iron deficiency anemia, there's three stages. Okay, stage 1, stage 2, and stage 3. Okay, in a blood test, these are the sort of things you would measure: hemoglobin levels, serum iron, okay, total iron binding capacity, and ferritin. These are the sort of things you would measure. Okay, now during stage 1, when we look at, so this person is developing anemia, stage 1 anemia. If you look at the blood test results, everything is normal, N for normal. Okay, so hemoglobin is normal, serum iron is normal, total iron binding capacity is normal. Okay, but what if you look deeper still, you would find that ferritin levels would be dropping. Okay, that's because there's just not enough iron, so the iron is being lost from the cells. Okay, but that iron that's being lost from the cells is being used to produce red blood cells and produce produce hemoglobin. Okay, that same iron is being used for transferrin to bind to, and so when we do a blood test, all of these things look normal. Hemoglobin levels, because the iron that's being lost from the cells is being delivered to the transferrin, that's being then delivered to everywhere else in the bone marrow for erythropoiesis. So everything looks normal. So when you do a blood test, the doctor would say, well, look, everything looks normal, nothing to worry about, off you go. Okay. Or the doctor could say, tell you what, why don't you come back in a few months' time? We'll see what happens. We'll see if there's a drop further still. Okay. Because another thing you have to consider is that red blood cells have a lifetime of 4 months. Okay, so the doctor might say, come back in 4 months' time. Let's see. We'll do another blood test in 3 or 4 months' time, and let's see what we get. Okay. So looking at, sorry, let me just go back. So stage 1, there won't be evidence of anemia at stage 1. Okay, not unless you do many other tests. Okay, so stage 2 is really leads to the exhaustion of iron stores. Okay, this is when you start to see uh, you, you you haven't quite got there yet. You haven't quite got to anemia just yet. Okay, but you start to see signs of it developing into anemia. Okay, so what you'll find is that during stage one, all the ferritin ferritin stored iron has been diminished. Okay, and that was sufficient to produce enough red blood cells to keep everything going, ticking along. Now, what's happened at stage two is that the iron transport compartment, so all the iron that's stored complexed with transferrin, that's being used up now. Okay, because that's being used up. Okay, the amount of hemoglobin is still normal because the iron that was being being transported as transferrin for the transport compartment. Okay, that was being used to produce hemoglobin. So hemoglobin levels will still look normal. Okay, serum iron will drop. The total iron binding capacity will start to increase, and what this basically means is that there's all this transferrin floating around in your blood, but there isn't enough iron for it to be there isn't enough iron for it to complex with. So the total iron binding capacity increases because there's all this extra capacity that's there, but not enough iron. Okay, to take up all the binding sites of transferrin. Okay, so the total iron binding capacity has increased. Okay, and if we look at ferritin, that there was a drop in stage one. Okay, that drop gets bigger in stage two. Okay, and it's not until we get to stage three. Okay, this is what we consider as frank anemia, stage three. Okay, this is when basically everything has run out. Okay, so the iron storage compartment, the transport compartment, and even the functional iron compartment is running out as well now. Okay, and because there isn't enough iron now in your whole body to recycle or to make up new red blood cells, your red blood cells that are being produced are basically not very effective because this is normal red blood cells, and these are um, red blood cells that arise due to frank anemia. You look at the phenotype of red blood cells here; they look very small. Okay, so they're microcytic, and they're not very effective. They don't work very well. Okay, so this is frank anemia, stage 3. Okay, and this is when you start to see. Now, when you see your red blood cells, if you have frank anemia or iron deficiency anemia, not all your red blood cells will look like this. Some will be normal size, some will be small, but the distribution width would be quite big. Okay, so that means you have a lot more small cells and a lot more normal cells, and so the distribution width will be bigger, and that does indicate, and that no, not that it does, but it can indicate iron deficiency anemia. Okay. Now, vitamin B12. Okay, so this is a very important nutrient. It's something that I've talked about in previous slides. Okay, as well as folic acid. Okay. Now, both of these are involved with DNA synthesis as well as synthesis of other molecules as well. Okay, and I'll talk about that in a minute. Just to be a bit technical, vitamin B12 is what everybody calls it, but scientifically it's referred to as cobalamin. Okay, and folate, also referred to as folic acid. Okay, so these are essential nutrients. It's not something we can. It's not something your bodies can produce by themselves. So they have to be taken from the diet. Now, vitamin B12. is a coenzyme, so it's something that's needed by other enzymes to help those enzymes work properly. Okay, so vitamin B12 is an important component of this enzyme here, methyl malonyl mutase, as well as methionine synthase. Okay, so both of these enzymes require vitamin B12 as a coenzyme. Okay, so if there's not enough vitamin B12, these enzymes don't work, and these enzymes are important, as you'll see in a minute. Okay, folic acid is actually a substrate for the conversion to what we call tetrahydrofolate. Okay, and this is needed for the production of this nucleotide here, thymine. Okay, thymine. So remember, there's 4 nucleotides: A, T, C, G, and adenine, thymine, cytosine, and guanine. Those are the 4 nucleotides that make up DNA. Okay, so if you have a deficiency of thymines, you simply can't produce DNA at the right rate. Okay, because your DNA, as it's being synthesized, is looking for a T, for a thymine to be added, and it can't find it because there's simply just not enough floating around. Okay, so DNA synthesis slows down. Okay, so folic acid is required for effective DNA synthesis, and in fact, vitamin B12 and folic acid both integrate. They have separate pathways, but both pathways integrate. Okay, and it's only when you have sufficient amounts of both vitamin B12 and folic acid that you get effective DNA synthesis. Okay, so this is where this is the role of vitamin B12. So I said that the vitamin B12 is a coenzyme for both methyl malonyl mutase and methionine synthase. Okay, now cobalamin itself, or vitamin B12, has to be converted to adenylyl cobalamin in order to be an effective coenzyme for methyl malonyl mutase, or it has to be converted to so vitamin B12 has to be converted to methyl cobalamin in order for it to be an effective coenzyme for methionine synthase. Okay, so it's not a simple case of vitamin B12. That's it. End of story. There's other processes for it to be converted to other molecules. Okay. Okay. Um, adenylyl cobalamin, once vitamin B12 has been converted to adenylyl cobalamin, okay, it's then an effective coenzyme for this enzyme, which mediates this reaction, converting methyl malonic acid (MMA) to 6-phenyl-CoA. 6-phenyl-CoA is actually needed to produce porphyrin, or the protoporphyrin, for heme synthesis. Okay, so you can see that there's a pathway leading to hemoglobin synthesis, and vitamin B12 is required. Vitamin B12 is also required for detoxifying this molecule here, homocysteine. It's a molecule that can become toxic if its levels reach a certain, or if its concentrations reach a certain level. Okay, and so methionine synthase basically detoxifies this homocysteine by converting it into methionine. Okay, the interesting thing about methionine is that it's an amino acid. Okay, but it's one of those essential amino acids. It's one of those amino acids that your body can't make itself. It needs to be received from the diet. So it's an essential amino acid. It's also the amino acid that starts it also marks the starting point of a protein. Okay, so the first amino acid of any protein is a methionine. So you can see how important methionine is in the context of protein production, but also how important vitamin B12 is in the production of methionine, but also in detoxifying your body from homocysteine. Okay. Now, in a blood test, you can measure B12 deficiency, because if there isn't enough B12, okay, this reaction doesn't proceed, and so MMA builds up in the blood. Okay, there's a reference range, and if it goes beyond this, if it goes beyond this reference range, it could be that there's a B12 deficiency. Okay, that's one way of identifying or indicating whether there is a B12 deficiency by looking at the concentration of MMA in the blood. As far as folic acid is concerned, okay, you don't have to learn all these different processes. The main thing is really to understand that folic acid is received from the diet. It's absorbed by your cells of the small intestine, of the or the cells that make up your intestines, the enterocytes. Okay, and the folic acid, okay, is actually it undergoes a series of reactions, okay, to produce this active substance here called 5-methyl tetrahydrofolate. Okay, and it's this tetrahydrofolate that's actually used for DNA synthesis or to mediate the process of DNA synthesis. Okay. Okay, so let me just show you the reaction. Okay, so this is homocysteine. Okay, this is the tetrahydrofolate that's been converted from folic acid. So this is the methyl tetrahydrofolate. Okay, and basically, in order to detoxify homocysteine, what we need to do is we need to add a methyl group to it. So we exploit the methyl group from the tetrahydrofolate, okay, remove the methyl group and attach it to this homocysteine to make methionine. Okay, and it's used by this enzyme here, methionine synthase, also known as homocysteine methyltransferase. Okay, and it needs methylcobalamin as the cofactor, which is obviously a metabolite of vitamin B12. Okay, so you can see how tetrahydrofolate and vitamin B12 integrate not only to produce this very important essential amino acid, okay, but also this tetrahydrofolate, which has lost its methyl, then becomes effective, okay, to be used in the synthesis of this nucleotide here, thymine, which is obviously the building block of DNA. Okay, so you can see the integration of B12 and folic acid here. Okay, so just a bit more detail here, but just it's kind of showing the same thing really. You can see that the tetrahydrofolate comes in, okay, and folate is converted to, so this 5-methyl tetrahydrofolate, this methyl group is removed to produce methionine, leaving you with tetrahydrofolate, and tetrahydrofolate then undergoes a series of reactions where it can participate in the production of thymine. Okay, thymine, which is then used to manufacture or synthesize DNA. Okay, so you can see over here, bottom line is this, that vitamin B12 and folic acid is therefore essential for DNA synthesis. Okay, and if you have a deficiency of vitamin B12 or folic acid, it will have an impact on DNA synthesis. Okay, now that's important because, like I mentioned before, when you think about red blood cell manufacture, although mature red blood cells don't have nuclei, so they don't have DNA, but the precursors that make up red blood cells do have DNA. They do have a nucleus. Okay, and if you want to produce more red blood cells, you need to produce more precursors. More precursors lead to more red blood cells, and those precursors have nuclei, have DNA in them. Okay, so if there's a drop in these nutrients, okay, we simply cannot maintain the rate of DNA synthesis that's required in the bone marrow. So you get a drop in precursor number, and therefore you get a drop in red blood cell number. Okay. Okay. So DNA synthesis is like I mentioned before. Okay, so like I said, DNA synthesis is really important in cells which are reproducing or dividing quite quickly. So skin cells are rapidly dividing. Okay, the cells in your mouth, the epithelium in your mouth, the epithelial cells which in your lungs, in your liver cells, in your gastrointestinal tract. These are the kinds of cells which are rapidly dividing. Okay, so new cells have to be produced, and so they're sites for rapid DNA synthesis. Now, your bone marrow cells are also sites of rapid DNA synthesis. So, okay, so your hematopoietic tissue, so the cells that make up all blood cells, are sites where cells are dividing rapidly. Okay, so if there's any impairment in of DNA metabolism, okay, which will happen if vitamin B12 and folic acid isn't consumed adequately, or your body finds it difficult to process these nutrients, and then it will have an impact on the rate of DNA synthesis. Okay, so and because DNA synthesis is important for erythropoiesis, it will have an impact on erythropoiesis. So it can cause anemia. Okay, so vitamin B12 deficiency or anemia because of vitamin B12 deficiency is rare, but it is more common in vegans or people who are strict vegetarians. Okay, and people who are strict vegetarians have to be or they have to take supplements of vitamin B12 because it doesn't exist in plants. Plants don't synthesize vitamin B12. Okay, meat and dairy sources are the only sites of or are the only sources of vitamin B12. Okay, the good thing about vitamin B12 is that it's not heat labile, which means that it doesn't diminish during cooking. Okay, so you can cook it, you can apply heat to it, and it'll still stay active. Okay, that's unlike folic acid. Folic acid, as soon as you heat it, it gets destroyed. Okay, um, uh, not all of it gets destroyed, so a little bit remains behind. But if you want to get as maximum folic acid, it's best to eat those plant sources which are rich in folic acid, to eat them raw. And you will need more vitamin B12 at certain times in your life, especially if you're a woman who's pregnant. Okay, so there's an embryo growing. Okay, new cells have to be produced to make the growing child, and so we need more vitamin B12. Okay, during production of milk. Okay, your milk is fortified with iron. Your body ensures that more iron is available to the child through the milk. Okay, and generally during growth. Okay, so more vitamin B12 is required during growth. Okay. Now, the whole the whole process of vitamin B12 absorption requires a multitude of proteins, a multitude of events, and all of those events to happen in a coordinated way. Okay, and this is what I'm going to describe now. Okay, so the absorption process. Now, as I said, vitamin B12 comes from your diet. It's not something that your body is able to produce. Okay, the thing with vitamin B12 is it's usually complex with other molecules, and in order to absorb vitamin B12, we need to detach vitamin B12 from those other bound molecules. Okay, and so in order to do that, what happens is that once the vitamin B12 enters the stomach, your stomach releases, not only does it release acid, hydrochloric acid, but it also releases an enzyme called pepsin. Okay, it starts off as pepsinogen, but then it gets activated by the acid to become pepsin, and that pepsin then allows, or it breaks the bond holding um, vitamin B12 with the other molecules. Okay, so pepsin breaks up this molecule here to release the free vitamin B12. So I'm saying vitamin B12, but it's abbreviated as CBL here, cobalamin. Okay, so it means the same thing. Okay, and once the vitamin B12 has been detached from its original partner, okay, it then becomes bound to another molecule called haptocorrin. Okay, it's R in the diagram, so this is haptocorrin. Okay, so this originates from the saliva. So when you consume something, obviously there's saliva mixed with the food. It gets into the stomach. That's inside the saliva would be haptocorrin. That haptocorrin then would bind to vitamin B12. Okay, and so this is just two steps so far, but both of them have to happen in a coordinated way. Okay, and then the next step, okay, is the release of this very important molecule called intrinsic factor. Now your parietal cells in the stomach, which also release hydrochloric acid, but they also release intrinsic factor, a very important molecule. Okay, um, the intrinsic factor actually binds to the vitamin B12. Okay, and that binding of intrinsic factor to vitamin B12 is essential for absorption of B12 into the cells that make up your small intestine, or your intestines in general, the enterocytes. Okay, B12 dissociates from the intrinsic factor. Okay, inside the cell. Okay, so it needs the intrinsic factor bound, and only when it's bound to the intrinsic factor can vitamin B12 be absorbed. But once it's inside the cells, okay, the intrinsic factor comes off. Okay, B12 then gets bound to transcobalamin, TC. Okay, so intrinsic factor detaches from vitamin B12. Vitamin B12 is then free to bind to transcobalamin. Okay, and is this vitamin B12 complex with transcobalamin then enters the blood circulation and eventually gets to the liver where it's stored. Okay, so you can have large quantities of vitamin B12 stored in the liver. Of course, that storage gets used up, and if you're not consuming enough vitamin B12, then it can become empty. Okay, okay, okay. Now, so to explain or to help explain why vitamin B12 won't be or isn't absorbed in some people, okay, it could be because of any of those stages. Okay, it could be due to a failure to separate B12 from food proteins. Okay, now if a person has gastritis, okay, they could be um, overusing alcohol or taking too many medicines. Not necessarily taking too many medicines. They could be prescribed for you, and you could be using them as your doctor has prescribed. But um, one of the impacts of, especially of aspirin, is that you can develop, if overused, can develop gastritis, and sometimes you can get medicines to be taken with the aspirin to prevent gastritis. Okay, so you have a multitude of medicines. Um, one medicine to treat the actual, problem, and then other medicines to treat the side effects of the first medicine. So sometimes it can be a mess. You can often see people with a tray of medicines that they have to take every single day, and it's usually when you get a bit older. Um, have many conditions, and then to treat those conditions, you have many drugs. Those drugs will have side effects. Then you need to take other drugs to treat those side effects to maintain some quality of life. Okay, but anyway, um, okay. So if you have gastritis, the cobalamin or the vitamin B12 doesn't detach from its original partner. Okay, and that's it. If that doesn't happen, well, full stop. Okay, it doesn't matter what you do after that. It's just not going to be absorbed. Okay. Uh, okay. But if that does happen, then stage two, where the where the vitamin B12 has to detach from haptocorin, maybe that doesn't happen. Okay. Again, that can be also due to a lack of gastric activity as well. Okay. And then another thing is intrinsic factor, this very important protein molecule that's needed to bind to vitamin B12 in order for it to be absorbed in the intestines. Okay. Um, if you don't produce enough intrinsic factor, you can't absorb vitamin B12. Okay. And then there's just simply other bowel diseases such as celiac disease. Okay, that can cause malabsorption. Okay, irritable bowel syndrome, celiac disease, so these gastrointestinal problems. All of these can have an impact in the ability of your intestines to absorb vitamin B12. Okay, now I've highlighted this one in red, intrinsic factor, because this is what I'm going to talk about next. Okay, the lack of intrinsic factor leads to what we call pernicious anemia. Okay, now pernicious anemia is actually an autoimmune condition, which means that it's your own immune system which is causing the problem. Okay, now if you remember, your parietal cells, which release hydrochloric acid in your stomach, they also release intrinsic factor. Okay, now for some reason, your immune system over time, it only happens to a small number of people, so it's quite rare, but in some people, your immune system over a long period of time, and this usually happens sort of like when you're in your sixth decade of life, so when you're like 60 or 60 plus, um, 60 plus years of age. Okay, this is when we actually can see pernicious anemia developing. Over time, what can happen is that your immune system can, for some reason, recognizes your parietal cells as being foreign. Okay, it recognizes that as them being foreign, and it destroys those parietal cells. So remember, those parietal cells are important for making intrinsic factor. So those cells are destroyed, and you don't get the production of intrinsic factor. Now, it doesn't mean that your immune cells will destroy all your parietal cells. Okay, it'll destroy some, and there are millions and millions. It'll destroy some, but it basically means that you're not producing enough intrinsic factor. Okay. In terms of a laboratory test to test this pernicious anemia, because it's an autoimmune condition, we can do an antibody test. Okay, so can we see antibodies against intrinsic factor? Okay, we can detect those antibodies that bind to intrinsic factor, and we can detect antibodies that bind to parietal cells. Okay, usually 90% of the cases um, are, is where antibodies bind to parietal cells. Okay, so we can diagnose that problem kind of straight away, because we know that your body is producing antibodies towards your own parietal cells. And parietal cells are responsible for producing intrinsic factor, and intrinsic factor is needed to absorb vitamin B12. Okay. Okay. So um, another type of anemia, again, this is extremely rare. Okay, is aplastic anemia. This is when really there's a failure there's a failure in bone marrow. Okay, so your bone marrow fails. All the cells that make up your bone marrow fail. Okay, and because erythropoiesis happens in your bone marrow, there's a reduction in red blood cells. Usually with aplastic anemia, what we have is because it's a failure of all bone marrow, and bone marrow is used not only to produce red blood cells but also to produce platelets and white blood cells. What we get as a result of aplastic anemia is not only do we get a reduction in red blood cells, but also a reduction in the other types of blood cells, such as platelets and white blood cells. And when we get a reduction in all three types, we call that pancytopenia. Okay, so a reduction in all three classes of bone marrow cells is called pancytopenia. Okay. Okay. Sickle cell anemia. Okay, so um, now sickle cell anemia belongs to an umbrella of conditions called hemoglobinopathies. Okay, hemoglobinopathies, of which the biggest one is sickle cell anemia. Okay, and it's usually because of there's structural defects in hemoglobin. Okay, so hemoglobinopathies are diseases where there's structural defects in the hemoglobin. Okay, now all of these globinopathies rather arise due to mutations in the genes that are involved with the synthesis of hemoglobin. Okay. Now, the most common hemoglobinopathy, like I said, is sickle cell disease. Okay. And the mutation that leads to sickle cell is what we call a point mutation. So it's the changing of one single nucleotide, and by changing one single nucleotide, you're changing uh, the outcome of which amino acid is produced. Okay, so let me just show you that. Before I show you that, the mutation that leads to the production of hemoglobin in sickle cell disease leads to HbS, hemoglobin S form. Okay, so it's a different form of hemoglobin that's produced. How that comes about is, like I said, it's a point mutation. So this is just one codon. Remember, a codon is three nucleotides, which codes for one amino acid. Okay. Now, among those three nucleotides, only one of those nucleotides has been mutated. So rather than a GAG, it becomes a GTG. And a GAG codes for a glutamate, whereas a GTG codes for a valine. Now, a glutamate is electronegative, which means it has a negative charge. Okay, whereas valine doesn't have a charge. Okay, so that has a charge and that doesn't have a charge. And simply by making a change of this one amino acid, remember this is a big molecule. It contains hundreds of amino acids. Okay, by just changing one amino acid, it completely changes the structure of the hemoglobin when it's deoxygenated. Okay, so when it's deoxygenated, this mutation causes hemoglobin to adopt an abnormal shape in the absence of oxygen. So when oxygen isn't bound, so it's deoxygenated, rather than existing as individual molecules or individual sort of units of structure, it then this hemoglobin start to bind to each other to form a fibrous protein. Okay, and that fibrous protein, that fibrous hemoglobin, causes the red blood cell to adopt this sickle cell shape. Okay, so it can't produce this nice rounded semi-concave shape, but rather this sickle cell shape. And this is not very effective in absorbing or binding and releasing oxygen. Okay. Okay, so in the absence of oxygen, um, the hemoglobin conforms to this fibrous structure. which leads to the formation of this shape of red blood cells. Okay, so here's a picture of a normal hemoglobin. So you can see individual units of hemoglobin. Okay, and this is hemoglobin S. So this is the mutated form of hemoglobin, which gives rise to sickle cell. The problem with sickle cell disease is not only are they ineffective really in distributing oxygen properly, but they don't have the same flexibility as normal red blood cells. And because they don't have the same flexibility, they tend to get trapped in very small capillaries. They get trapped, they clog up, and that can initiate the clotting cascade. So clots can happen. So people who develop sickle cell disease also develop clots, and that's obviously a concern because non-specific clot formation can have, if it happens in the heart, you can develop heart attacks. If it happens in the brain, you can develop strokes. Okay, so very, very dangerous condition. We've talked about anemias now. So a drop in red blood cell number. Now let's talk about conditions where there's excessive numbers of red blood cells. Okay. Now these two terms, polycythemia and erythrocytosis, are sometimes interchanged, but they shouldn't be because they mean slightly different things. Okay. Um, both polycythemia and erythrocytosis leads to an increase in erythrocyte number. So that's what they have in common. Okay. In both cases, what you'll find is that the red blood cell number has increased. Okay. Now, when you have an overrepresentation of red blood cells, that can cause your blood consistency to change. It becomes more viscous. Okay. And there needs to be a certain level of fluidity for blood to perform its functions. And so if it becomes viscous, okay, to move this less viscous fluid around blood vessels, you need to generate a bit more pressure. And so therefore your heart has to work harder. Okay, so the heart has to work harder, it has to pump more blood, it has to generate more pressure. Okay, and because it has to generate more pressure, okay, that can increase the chances of clots developing rather. Okay, and if those clots happen obviously in the brain, it can lead to a stroke. Okay, so maintaining the right viscosity or the right fluidity in your blood is really important to prevent these types of conditions from happening. Okay. Okay. So, like I said, both polycythemia and erythrocytosis show increased levels. So, where the hematocrit levels are much higher than the normal reference range. Okay. So, although erythrocytosis and polycythemia show very similar results, the pathologies are quite different. And let me just explain what I mean by that. Okay, so this is to show the difference between polycythemia and erythrocytosis. Okay, so polycythemia is the condition in which both the red blood cells and hemoglobin levels increase above the normal levels. Okay, and what this basically means is that the red blood cells look normal, but the numbers have increased, whereas erythrocytosis, okay, is that the red blood cell mass increases beyond the normal level, but the hemoglobin levels don't necessarily have to change, which means that in many cases with erythrocytosis, the red blood cells um, can be quite small in some cases. Okay. Now, both polycythemia and erythrocytosis can be quite normal conditions. Okay, if somebody suspects polycythemia or erythrocytosis, what you do is you do a blood test, you suspect it, you see the results, and the results indicate that there is polycythemia or erythrocytosis, you do another blood test a couple of months down the line, and let's see whether you get the same result. Okay, so if it's polycythemia and you suspect it's cancer, it will be cancer 2 months down the line. Okay, it'll be cancer 4 months down the line. Okay, so what we do then is we do another blood test, and if it's still if there's still high numbers, then you've kind of confirmed polycythemia. Of course, you don't have to wait to confirm it. You can do other tests to um, to ensure that to confirm um, your diagnosis. Okay. Okay. Now, with polycythemia, because usually it's a genetic problem, it's cancerous. There's a problem in the bone marrow. Okay, so there's some kind of genetic problem, and it's a problem in the bone marrow. Remember, the bone marrow gives rise to not just red blood cells, but other blood cells. So what you'll find with polycythemia is that not only do the red blood cell number go up, but also the white blood cells and the platelets may also go up as well. Okay, it just depends where the problem, where the mutation is affecting. Okay, whereas with erythrocytosis, and always with erythrocytosis, will only the red blood cell number go up. Okay, so that's the difference between the two. Okay, now with erythrocytosis, if you suspect erythrocytosis, sometimes the simplest explanation is usually the correct one. Okay, it this is a normal blood sample. Let's say that's the plasma, that's the red blood cell volume. Now, if somebody is dehydrated, remember the plasma is mostly water. So if you're dehydrated, your blood plasma will look um, like this, and your red blood cell volume will look like this. But if you look at the red blood cell volume, it's the same as normal. Okay, so in fact, there is no erythrocytosis here. The red blood cell fraction is still the same. You're still getting 40%. In terms of the not 40%, but you're still getting the same kind of concentration of red blood cells. It's just that the proportion of it in relation to the plasma is much more. So it looks like there's more. Okay. So if it is due to simply dehydration, we call that relative erythrocytosis because it's relative to the loss in plasma. Okay. And it's not because there's an increase in red blood cell number. Because it's the same as a person who's considered normal. Okay. Uh, okay. So just to remind you, before I move on to the next step, is that erythropoietin is the hormone that increases the rate of erythropoiesis. Okay, and erythropoietin is expressed when hypoxia is detected. So you get increased levels of EPO, erythropoietin, when hypoxia is detected. Okay. Now, hypoxia can be detected, or hypoxia can occur in people who have their lung conditions mismanaged, or let's say they have COPD, chronic obstructive pulmonary disease, of which there is bronchitis and emphysema, and also severe asthma as well. Okay, usually there's inflammation, there's remodeling of your lungs, which basically means that the exchange of gases, or the rate at which gases are exchanged in your lungs, so the oxygen going from your alveoli into your blood, and the exchange of carbon dioxide from your blood to the alveoli, the rate at which these gases exchange is reduced. Okay, so there's not enough oxygen being delivered to red blood cells. Those red blood cells, they don't have a they don't they, obviously then they can't deliver the right amounts of oxygen to other cells. So hypoxia can develop in those situations. Okay, so in those situations, then what you'll find is that the concentration of EPO will increase, and as it increases, the rate of erythropoiesis will increase, and that leads to what we call absolute erythrocytosis. So this is where the actual number of red blood cells is actually higher than what it needs to be. Okay. Okay, so just to kind of finish off now, another reason for polycythemia, not another reason, but one of the main reasons for polycythemia, or where there's an increase in the number of red blood cells, is simply because there's a failure to regulate the number of red blood cells. Okay, so imagine the situation where a certain level of erythropoietin is produced, and you get a certain level of red blood cell manufacturing going on. Okay, so this is how it would normally work. Erythropoietin would bind to the erythropoietin receptor. That receptor would then signal or initiate a series of events inside the cell, which leads to an upregulation of those genes responsible for the manufacturing of red blood cells. Okay. Now, what happens is that when there's enough red blood cells, your body will detect that. Okay. And it'll shut down this pathway. Okay. It'll shut down the pathway through negative feedback. Okay. So it'll shut it down, even though there's erythropoietin knocking about. The system is shut down, so it won't lead to the production of any more, or the activation of any more genes that leads to the production of red blood cells. Okay, so that's the normal situation. Now, so there's a negative feedback regulation mechanism here. With polycythemia, okay, there's this protein here. It's called JAK2, and in the normal situation, this JAK2 works normally. It responds to negative feedback, but the mutant form doesn't. Okay, it doesn't respond to the negative feedback, and so when your body says enough red blood cells, no need to produce any more, the protein simply doesn't respond to it, and so there's no negative feedback, and so your cells continue to produce more red blood cells. Okay, so, and that's obviously the reason for cancers. Okay, or cancers of at least of the blood. Okay, so that's it. Okay, I don't really want to say anything other than that. Uh, Although I will say is that just make sure that you you learn all the different concepts. Okay, there's a lot to learn there. Okay, go through it step by step. Take some notes, and just go through it over and over again. Okay, because everything other than that one little bit that I said, don't learn this. Okay, make sure you do learn everything, or as much as you possibly can. Okay, just go through this PowerPoint over and over again. Look at YouTube videos. If you don't understand concepts, send me an email. Okay, but don't sit there hoping that it will come together at some point by itself. Okay, it is going to require hard work. Remember, this is level 5, so it is the next level up. Okay, which means you have to put that your effort levels one level up as well. Okay, but like I said, I'm here to help. Martin's here to help as well. If you have any questions, if there's anything that we can help you with, please don't hesitate. Okay, right then. I think at this stage I'll say that's enough then. Okay, so goodbye, and just to finish off, then there's a series of questions here. Please attempt these questions, okay, because they do give an indication of the types of questions that you might get in the actual exam. Okay, so go through the PowerPoint, see if you can answer these questions. If you can, if you can't, go through the PowerPoint again, But please don't cheat yourself. Don't look at the answers, which I will release in maybe in a day or two. Don't look at the answers and revise that way. That's not the way to revise. Okay. Right then. Anyway, that's enough of me jibber jabbering. So I'll leave it there. Okay. So until next time, goodbye.